with increasing attention on early manifestations of transthyretin amyloidosis, as well as emerging treatments. Appropriate studies to confirm the types and variants of transthyretin amyloidosis are fundamental to improve prognosis. The main advantage of this protocol is that this test is feasible and easy to perform in the clinical laboratory.
To begin the protocol, obtain a DNA extraction kit. Add 10 milliliters of solution 1 to the previously obtained 4.0 milliliter blood sample and incubate the sample on ice for 10 minutes. After incubation, centrifuge the sample at 2000 G for 5 minutes at 4 degrees Celsius. Carefully remove and discard the supernatant. Resuspend the pellet in 3 milliliters of solution 1. Centrifuge the sample at 2000 G for 5 minutes at 4 degrees Celsius again. Discard the supernatant. Resuspend the pellet in 3 milliliters of solution 2. Next, add 10 microliters of pronase stock solution to get a final concentration of 100 micrograms per milliliter and mix the contents well. Incubate the DNA at 37 degrees Celsius overnight. The next day, chill the tube on ice for 10 minutes. Add 0.8 milliliters of solution 3 to the sample and invert it 3 to 5 times.